What if we can peek in different cell types and see how the molecular machine that synthesizes all proteins in the cell differs? What if the ribosome changes during disease? What if we can detect defective ribosomes? What if we find out a way to help them clear those defective ribosomes? Can that help us slow down diseases such as cancer and neurodegeneration? It's funny, I feel like I've always wanted to be a scientist, but I didn't even know the word scientist. I grew up in Bulgaria, in a tiny village called Shipka that is high up in the mountains. I was raised by my mother and my grandmother. When I was growing up, there were certain norms of what a young girl should do or say, or what a career for a girl should be. I think it was very important that they were not repeated to me at home. I had this very magical childhood where I was just very free. I was reading about replication and protein synthesis. I was reading about ribosomes back in the day when I decided to apply for college. My mom never said, like, how would the Bulgarian girl from Shipka end up at MIT? She was just, of course you apply for college, of course you're going to be a scientist. Her belief in me drives me to today. I didn't take no as an answer. As a freshman, I was really excited about being in a lab and actually doing some of the experiments I was reading about. I emailed a few professors and they all said no. And that was a bit jarring for me because I came all the way from Bulgaria, right, with 2 bags and all my dreams in them. But I was very persistent. I sent lots of emails and I got 2 interviews and 1 lab took me up. I think I really fell in love with ribosomes towards the end of my PhD. We were studying how a ribosome, as it moves along an mRNA, it can encounter a problem. Like there's something on the mRNA the ribosome cannot deal with. But that doesn't make sense to me because how would a cell know that the problem is in the mRNA, not the ribosome? There must be a way for cells to actually find defective ribosomes and deal with them. And that was a question that I feel like no one else had asked before. And it was really exciting. After I finished my PhD, I started my own lab as a fellow at the Kamena Institute, and then I moved my lab here. We're building this very solid foundation on which future research can build upon. A lot of human diseases such as cancer and neurodegeneration are also accompanied by the accumulation of defective ribosomes. The really challenging question in the field has been how do we identify these defective ribosomes? You know, every single cell in your body has about 3 million ribosomes. So if a single ribosome is broken, it's like finding a needle in a haystack. We have developed unique tools to specifically label a defective ribosome and follow them in the cell. We also study how ribosomes are built. We decided to look at a very specific system, which is the developing oocyte. Oocytes are eggs. What we discovered is that the oocyte takes a very different strategy in building ribosomes compared to any other cell in your body. As the oocyte ages, its ability to build ribosome is altered. We're very curious if that can also have impact on fertility. I don't like putting myself or my science in a box. I don't like limits. Having this open mind and trying not to label your research or what you do is absolutely critical. We're not simply ribosome biologists, we're so much more, right? We can take the ribosome and study development, or you can study human disease or evolution and species diversification. I have a 5-year-old and one of our favorite things is cooking together. And I can definitely see myself in him.
It was a recipe in my head.
Oh, really? Does not follow a recipe. He always says, I have a better idea and I think we should do this way.
Wow,
I think what if questions are absolutely essential. They allow us to dream and they allow us to push the boundary and be creative. But we always keep in mind that these biological questions and the discoveries that we make need to impact human health. What if we figure out how cells can detect and clear defective ribosomes? How many types of diseases can that impact? With all the technology that we have today, science is moving so fast. What if we don't have to wait?